able to see my screen now?
Yes, your screen is visible.
Thanks, Aditya.
Yes.
So guys, welcome to the project session on day 4 morning session, which is on deep brain stimulation models for basal ganglia. So today, this is the overall agenda with which I have come up with. We can have ups and downs with the overall agenda. We can tweak it a little bit to accommodate others' questions as well. So overall today, since it's basal ganglia, it involves Parkinson's disease. So we'll be starting with a basic introduction of Parkinson's disease, and then we'll go through the basal ganglia architecture and what are these ST and GPE dynamics and why are they very important for Parkinson's. Then we will actually look at the ST and GPE network model and how do we simulate it under normal condition as well as PD condition, that is Parkinson's condition. Then we'll also have a look at deep brain stimulation of STN, which is actually a clinical procedure. I'll go through it very briefly during the session, and also a model of that deep brain stimulation method and how does it work. Then we'll actually have a full code simulation session for STN GPE dynamics alone under normal condition, Parkinsonian condition, and Parkinsonians with DBS conditions. With that, we will be sort of covering up the dynamical aspects of all the Parkinson's condition. Then we'll go to a little bit behavioral level. That is, we'll look at some kind of decision-making deficit, some kind of a gambling disorder that we see in Parkinson's patients. And we'll look at a brief reinforcement learning-based model of basal ganglia and use it to simulate that decision-making under normal PD and PDBS. So this is like kind of overall agenda for today's session. We are assuming like the first 1.5 hour session, we'll be trying to do the dynamics alone. And then remaining 1 hour of the session, we'll be going through the decision making. Okay. You guys can have a look at the paper after the session as well. I'm not going to go deep through the research paper itself. I'm only going to talk about brief stuff, which we are going to do today as a part of the project. But you can always come back to us talking about the paper and discussing about it. So starting with the introduction to Parkinson's disease, I'm sure a lot of you might have heard about Parkinson's disease. It's like world's second most predominantly, prevalent disease. It's actually a neurodegenerative disorder, which is actually mostly characterized by motor symptoms. Late phases of life, generally people get attacked by Parkinson's disease, and most of them end up showing motor symptoms. By motor symptoms, I mean tremor, rigidity, and bradykinesia. That is, you ask them to reach a target, they end up shivering a lot, which we call it as tremor, or their movements are very slow, which we call it as bradykinesia, or they get stuck at a specific moment, which is basically rigidity. And what is the physiological cause of it? It's because of death of dopamine cells, which is present within substantia nigra pars compacta in basal ganglia. You can think all of them as certain nucleus or certain clusters of neurons which are present deep inside the brain. Like basal ganglia is a full subcortical structure or a cluster of neurons which are present, which has another sets of cluster of neurons, which is SNC, that is substantia nigra pars compacta. Now, if we actually look at basal ganglia in itself as an input-output kind of a system, it generally has the input port through which all the information comes through in, which is actually a striatum. And there is an output port through which the information moves out, which is actually GPI, which is globus pallidus interna. I know these terms might be very new to you guys, but it's totally fine as far as the network diagram is always there for you guys to understand what is input and what is output. And apart from these two, input and output, there are some kind of intermediate structures, which are basically subthalamic nucleus and globus pallidus externa, STN as well as GPE. I highlighted them because the first half of today's session will be based on these two structures. We'll have a look at it in brief on literature review and then go through the simulations. So as I already mentioned, Parkinson's disease is majorly a motor symptom disease where you see slowness in movement, stiffness, they can't walk properly, etc. But apart from motor symptoms, there are also a lot of cognitive symptoms. I'm sure a lot of you might have seen people with a lot more cognitive issues at later phases. Parkinson's also does that. And one of the core deficit is decision making. Now, decision making is very, very important in daily life. It basically, like decision making can be as simple as choosing which food to eat at this moment. It can go up to a complicated task of which stock do you want to invest in. So it's very important and it's a very core component of daily life. And decision making gets disrupted in Parkinson's patients, which we are going to look at towards the end of the simulations today. Like towards the end of the session, we'll actually look at one decision making task which people use to assess PD. Going through the next part of the session, that is, what is the basal ganglia architecture and what is the importance of STN-GPE system? Till here, if anyone has any queries, if anyone joined late, kindly let me know. If not, I'll go ahead with this. Okay, maybe I'm assuming that there are no doubts still here, so I'll go through the front part and further. Going through the basal ganglia architecture, this is how a couple of books show up how the basal ganglia architecture or how the connectivity across the structures look. Though it looks very complicated, you can put it as a network diagram very simply. That if there is any kind of input, it can be sensory input or a visual input, etc., that goes through the cortex, that enters the input structure. As I said, input structure is striatum. Now, striatum also has two parts to it. One we call it as D1 striatum, and the other we call it as D2 striatum. D1 and D2, think of it like an input port into the system. Any network will have an input. So the D1 and D2 act as the input to the basal ganglia. Now, from here, there are two pathways that split into. One, we call it as a direct pathway where the input directly connects to the output, which is GPI. And the other is an indirect pathway which actually goes through the system, STN-GPE system, and connects to the output. From output, it gets connected to the thalamus. Any issues still here? So basically, overall basal ganglia, you can think of it like there are two pathways. One is direct, which connects input directly to the output, and the other is the indirect pathway where it connects via the via STN-GPE system. Cool. Yes. Okay, moving ahead with this. Now, I have highlighted the direct pathway in the structure here. That is, input goes to cortex, cortex to the input structure, input to the output structure, from which it goes to the thalamus. Can you guys think about what would be the overall effect of direct pathway in a way? Assume that the connection from input to GPI, that is output, is inhibitory. From there to thalamus is also inhibitory. What do you think will be the overall effect of direct pathway? Anyone? Sorry, I can't see the chat.
There's a chat answer.
Excitation of thalamus. Yes. Not excitation at the thalamus level. Yeah, I mean, the overall effect is basically, yeah, it would be a positive effect because I'm having two negative. Negative into negative will become overall as a positive effect. That is it. So that is the reason we call it actually as a go pathway. That is, it promotes movements. Similarly, if you go through the indirect pathway, this is the overall indirect pathway I'll have. I'll have a negative connections from striatum to GPE, GPE to STN. Again, it's a negative connection. STN to GPI is positive, GPI to thalamus is negative. What do you think will be the overall effect? I mean, it's little obvious now. Inhibition, yes. So basically, the overall effect of the indirect pathway is basically inhibitory in nature. That is, it's no go. It tries to prevent the movement in itself. That is the reason we actually call the direct pathway as a go pathway and indirect pathway as a no go pathway. Basically, because of the way the connections work. Now, as I said, in Parkinson's, dopamine cells are very much important. What happens when dopamine is low and what happens when dopamine is less? When dopamine is low, basically the dopamine has a kind of differential effect on the striatum D1 and D2. When there is a higher amount of dopamine, then it is fine. The direct pathway is higher and it's fine. The movements are smoother, you can move very easily. But whenever there is a lower dopamine, the indirect pathway becomes pretty high. What is the expected thing then? Then it has a lot of no-go moments. That is, the moments get stuck a lot. Since the indirect pathway is like high, you will be ending up having a lot more no-go kind of a movement, which is actually what is seen in Parkinson's patients. Parkinson's patients have this lower dopamine because of which the movements get stuck, or because of which your indirect pathway is higher, because of which your movements are stuck. And this circuit imbalance is what is the issue in Parkinson's disease. Now, since I already mentioned that indirect pathway is overactivated, what exactly happens to the STNGP dynamics per se in Parkinson's conditions? Any guesses, or anyone has any idea on what might be the case? I mean, one thing is for sure that since the indirect pathway is pretty high, obviously STN will have, yes, more inhibition to thalamus, and at the level of STN-GP dynamics, it will be overactivated, right? At the level of STN, it should be overactivated because then GPI inhibits the thalamus more. So that is what actually happens with respect to the STN-GP dynamics. That is, at the level of STN-GP dynamics, they will be overactivated under Parkinson's condition. So generally, if I take this as STN and this as GP, the way the connections work are like this. GP receives input from its D2 striatum or whatever the input is, which is inhibitory. Now, GP connects to STN with the inhibitory connections. Similarly, STN is excitatory. In fact, STN is the only excitatory nucleus that is present within the basal ganglia. STN connects back to GP with a positive kind of connections, that is excitatory connections. And apart from this, GP also connects within itself, which we call it as lateral connections. Similarly, STN also connects within itself. It's like this. There are two clusters which are connected with each other. That is, cluster A connects to cluster B connects to cluster A. And within cluster A also there are connections, and within cluster B also there are connections. Now, what exactly happens in PD when there are low dopamine that is present? When there is low dopamine present, STN becomes extremely overactive because this inhibits GP. So GP is supposed to inhibit STN, but since GP is inhibited, STN gets overactive. Okay. Now, due to this overactivity, first thing we see is the striatal currents are increased. Obviously, the D2 pathway gets activated more and more. Apart from that, there is also lateral connectivity that gets increased in STN because of dopamine. It's a kind of second line connections from dopamine. Dopamine also connects to STN and GP in itself, where the lateral connectivity gets very strong. Due to these two phenomena, STN ends up bursting. Do you guys remember what is bursting basically from your day one? Anyone who remembers what is bursting or chattering? You can type on your chat boxes, or you can unmute yourself and speak about it. Yes, not at large frequencies per se, but yeah, you're right to some extent. It's basically firing for certain time and then going silent, firing for certain time and then going silent. I'm sure you might have remembered that there is a continuous fire, then there will go silent, and again there's fire. It's like bursts, sudden spikes and then go silent, sudden spikes and then go silent. Yeah. So that is what actually happens under PD condition. Under PD condition, STN, which is like a continuous firing, goes from continuous firing towards under PD, which is more of bursting kind of a behavior. Bursting, not just bursting, they end up bursting around 15 to 35 hertz. And this beta frequency is very common across the literatures of Parkinson's. And people have been studying how does beta frequency influence motor movements, how does beta frequency influence cognitive stuff and all. Any issues still here? Okay. So this is what I meant. If you actually go inside the STN, keep an electrode to one single neuron and actually record it, you will see that spikes are clustered. Like these all are clustered together. Then again, they went silent for a while. Then again, they'll be clustered together. Again, it'll go silent for a while. Then again, clustered together. Then again, silent, then clustered. This is how actually the single neuron firing technically looks like. Though it's a little noisy one because it's an experimental recording, it will be noisy. But yeah, this is how they fire if you look at single neuron level. But now, apart from single neuron level, we can also record the activity from cluster, right? If I have some group of 100 neurons, I can record the overall activity of those 100 neurons. That is what we call it as LFP, or local field potential. You will actually see LFPs when you're doing the simulation. That looks more or less like this. And if you take the frequency spectrum of it, it actually shows peaks around some 15 hertz and some 25 to 35 hertz. This is what we called it as beta frequency. This 25 to 35 is beta. I think 15, around 15 to 35 is what we call it as beta. Any issues still here? Any doubts on what is an LFP basically?
Hello, ma'am.
Yeah, yes.
I just have a small doubt, like how actually you collect this information. Is it maybe from an EEG, or how you actually like this
You mean
information?
STN information?
Yeah.
So STN is located very deep inside the brain. It's not on the superficial level. So EEG doesn't capture it. They
Okay, so
actually
you
do a surgery and put
in,
electrode inside the brain
okay,
and then only record.
okay. So
Yeah.
you need some invasive type of mechanism
Yeah.
to
Yeah.
capture.
It
Okay,
will be an invasive mechanism. Yeah.
okay. Thank you.
Generally, people do it when they are doing deep brain stimulation because,
Okay,
anyways, then they have to actually undergo a surgery invasively inside the brain. They record it at the same time.
okay. Thank
Yeah.
you.
I have a doubt actually.
Yeah.
The local field potential is caused by the spikes, or is it by EPSP, IPSP, something like that? Or is it the
It,
burst spikes that cause it?
uh, local field potentials is from the EPSP, IPSP only.
Okay.
It's a voltage kind of a thing. So when I insert an electrode, when there are cluster of neurons, it will capture some kind of current from all of them together, right?
Okay. And this
That will give
beta
you electrode.
frequency is due to those activity, right? It has nothing to do with the burst, right?
So basically, since they are bursting in that frequency, see, if most of my spiking bursts are around 20 hertz, and if I take a cluster recording, then only I'll see that 20 hertz high rate. If no neuron is actually firing at 20 hertz, why will you see a local activity of 20 hertz or whatever frequency?
Okay.
Yeah. Any more queries still here? Okay. So moving ahead, we are going to look at the model of STN-GPE system. Till now, we are able to look at what are the experimental things of STN-GPE system, but now we'll look at the model of STN-GPE system. Okay, before that, I have a question. I want to build an STN-GPE model which actually shows these kinds of behavior, that is under low dopamine, where the STN becomes more active and synchronized bursting, and also normal condition. What do you think should be the model criteria should be? I mean, how do you start up building a model of STN GPE system? What is its behavior should look like? I mean, if I want to test the model, what do I look at it? How do I know my model is right? You guys can make any guesses. Anyone? I mean, you can just guess. Any random guess would be fine. Okay, so basically, whenever I have to build any model, I need to understand what are the criteria I need. Now, this STNGP network for sure should be able to show normal behavior as well as PD behavior as I keep changing some parameters. Don't you agree? Obviously, yes. The two things that I have to look at is as I keep increasing the D2 or the input current to the system, it should end up moving more towards PD kind of behavior. And similarly, if I keep increasing the lateral strength, it should end up behaving like a PD kind of a behavior. Yes. Is all of you guys on same page like that? Yes. So that is what we have to technically look at. That is, if I want to build a system, the system requirement should be like this. It should be capable of showing both normal as well as PD dynamics. When I say normal and PD dynamics, it is here. Normal means it's a simple continuous firing, continuous as well as desynchronized firing. I'll get into what is a synchrony very more clearly as we move ahead. And under PD, it should show such kind of a bursting phenomenon as well as the synchronized firing around beta frequency. And when does it show? If I keep increasing the striatal currents, that is at low current, I want normal behavior. At high currents, I want PD-like behavior. Similarly, at low lateral strength of STN, lateral strength is the connectivity strength within itself. That is, one STN neuron connects to other STN neuron with certain strength or with certain weight, synaptic weight. At that low weight, it should be normal, and high lateral strength, it should be PD. This should suffice the model requirement, right? Now, did you guys look at any kind of a model which showed both continuous as well as bursting behavior when you were looking at any of the neuronal models? I'm sure day one had a lot of neuron models. Any neuron model you saw which had, yes, Izikevich, right? So there are other neuron models as well, but we are sticking to Izikevich for the STN-GPE system. You're right. So we will be picking STN-GPE system to be an Izikevich neuron model. That is, this is how we technically use the model to be. I take 256 2D sheet of neurons of STN. Similarly, 16 cross 16, that is 256 2D grid of neurons for GPE. And both the neurons and all STN neurons as well as GPE neurons, both of them are Izikevich neurons. Okay. And then I will define how are they connected. GPE receives input currents from striatum, and GPE is connected to STN as well as within itself. That is, each neuron of GPE connects to some 70% of the other neurons within STN, and it also connects with itself with 70%. Similarly, STN also will have connections within itself and also connects back to GPE. Is this setup clear? That is, we'll have connectivity weights between GPE and STN to GPE, and also within GPE to GPE and STN to STN. This is how we do the connection. Now, I'm not going to go through each and every equation of STN-GPE system, but maybe I'll open up the paper to show you an overview. What is it happening? Maybe that would be better. This is the paper we have actually sent you guys. And this is the paper which actually has the STN-GPE network in itself. It's actually a huge spiking network, but I'm not going to go through each and every detail of it, but you will understand what I want to show. First thing is, as I said, both the neurons, each of the single neuron of 256 neurons in STN and GP, both of them are modeled as Izikevich neurons. If you actually see this, this is an Izikevich equation. Are you able to see that? Now, Izikevich equation has these two currents that comes into play. That is, one is the external current. Like if it's GPE, it receives currents from STN as well as striatum. Similarly, if it is, some kind of synaptic connectivity within itself or the lateral connectivity. Similarly, STN also has those currents. And each of those currents are actually modeled using their receptors, which for you guys, maybe it's not very much needed at this level, but at least understanding the overview that there is a spiking network for STN and GPE with connectivities made, that should at least suffice for today's lecture. Any issues still here? Good. Maybe then I'll also talk about how do we model the STN DBS, deep brain stimulation itself, and then we can go through the code simulations for you guys to have a hands-on on how does it look like. Do anyone know what is deep brain stimulation? You can let us know on chat boxes. Any idea on what is a deep brain stimulation? Yes, kind of. Yes, Vanika. Yeah, so basically, you're right. Deep brain stimulation is kind of a surgical procedure. As you remember, STN goes haywire, right? STN neurons start firing very rapidly and they have become pretty much overactive. One way people have started looking at it is if I send out electrical pulses and end up stimulating STN inside the brain, they thought that the overactivity of STN can be suppressed. That is what deep brain stimulation is. At later phases of Parkinson's disease, this is the only way to treat them. So the way they do it is they do a surgery, slightly open up brain a little bit. I mean, open up the skull a little bit, insert electrodes into the STN. Remember, STN is not superficially present. It's very deep inside the brain. So you have to do a surgical procedure irrespectively and ends up keeping an electrode like this inside the STN and start stimulating it using biphasic pulses. Biphasic as it for every positive pulse, there is also a negative pulse. The only reason we have such a positive negative pulse combination is there is no overall charge buildup. If I only keep giving positive pulses, there's a huge charge buildup inside the brain, which will be not good for cells. So they end up giving biphasic pulses. That is, I'll give a positive pulse and also a subsequent negative pulse so that there is no charge buildup. And this was sufficient for them to break through the synchrony. I mean, though not fully, but definitely a little bit of synchrony was reduced. You remember under PD, we saw a lot of synchrony, right? That can be reduced here. So this is what I meant by biphasic pulses. That is, for every positive pulse, there is a negative pulse. And generally, people tend to keep the frequency around 130 hertz. Okay. Now, whenever I want to define any kind of a DBS pulse in model or even when you're stimulating, I have to define what is its frequency, what is the amplitude of the pulse, what is the pulse to pulse interval, that is from this pulse to this pulse, what is the gap I need to have, and what is the duty cycle or the pulse width. You can think of it as pulse width. Duty cycle is like the fraction of it. But anyways, you can think of it as pulse width. These four parameters will define how my stimulus will look like. Even in model, or even if I want to actually give a stimulation, this is how I can measure it. Now, if I want to do a model of STN DBS, what I can do is I can take my 2D grid of sheet of neurons, place the electrode at some location in it. Say I'm putting it at the center, and then model the DBS. But my question is, when I keep an electrode at the center, do you think the influence at the center versus the influence to the corner, will it be the same or will it be reduced to the corner? That is, I keep an electrode at the center of the sheet versus to the corner. Will there be an influence reduction of the stimulation? Do you guys think that? Yes, right. This is what I meant when I say this is my STN, I keep an electrode at this neuron. Obviously, this neuron receives higher current in comparison with this one, right? Can you guys think of any kind of a distribution where you can actually have such kind of a drift? That is, as the distance increases, the influence will be decreased. Any kind of distribution? From the center, as you keep moving away, mathematically, which distribution will give you such thing? Gaussian. Yes, Shwanal. That is right. We technically do the spread as a Gaussian. That is, at the center, it will be high. And then as you keep going down, it will reduce. As you can see, the brightness increases, right? From red to it goes to blue. Blue is like the least, red is the highest. So we end up modeling the spread as a Gaussian. Now, this Gaussian ka standard deviation or the spread of it depends on your neuronal tissues as well. But yeah, so that gives us an overview of what is an STN-GPE model, how do we model it with respect to the normal and PD, and how can we do the DBS models. Any issues still here? If there are any issues, you can let us know now. If there are any doubts still here, otherwise we can go through the coding session. I mean, we can go through the model stuff.
May I know what is exactly lateral strength?
Lateral strength, yeah, sure. So basically, when I say strength, I have I have this neuron. This will be connected to other neurons, right? Within STN. I'm not talking about GP. They also have connectivity among themselves, and they'll connect with certain weight, right? It might not be one. It might be a weight of some 0.5, 0.6, 0.1. That is what is called as lateral strength. If that connectivity is very high, that is, all the neurons are very strongly tied to each other, then they will be synchronized. Does that make sense?
Also,
Yeah.
I have a doubt. This
Yeah.
STN overactivity in Parkinson's is associated with certain type of clinical features, right?
Yes.
I guess it is more like it is not associated with slowness, right? Like since it is making thalamus more active. What is the difference?
No. So basically, when you see the activity of neurons, when they become hyperactive, when I say hyperactive, all of them are actively firing around 15 to 35 hertz. Implies if all of them are firing at those frequencies, don't you agree? The overall amplitude of STN will be very, very high.
Yes, yes.
That if my STN amplitude is very high, it will overactivate my GPI, which in turn end up inhibiting my thalamus more and more. So it will give you no-go a lot more. So that slowness also will be kind of a behavior from that.
Oh, okay, okay, okay. Ha, ha, ha.
Yeah. So most of the motor symptoms are actually correlated to that kind of a frequency around. At least tremor has a lot more studies on it. Yeah.
Okay,
So
okay.
then we will go through the coding sessions of the STN GP system.
One more doubt actually.
Yeah, yeah.
So can you just quickly summarize the mechanism of that dopamine deficiency and how it leads to these two pathway problems? Just
Yeah.
to finalize that.
Yeah, sure. So basically, dopamine. So first thing we need to remember is the direct pathway is go, indirect pathway is no go in its overall effect. That is, if my direct pathway is overactivated, then it will always lead to a random movement. And if my indirect pathway is overactivated, it will try to stop the movement or slow down the movement. So when there is a lower dopamine, then the indirect pathway gets overactivated. That's how dopamine influences both D1 and D2 striatum. It gets connected to both of them. So when the dopamine is very low, the indirect pathway is overactivated. STN gets overactivated, due to which there is a lot of no-go. If you even not only motor movements, if you actually look at those tasks, I don't know if you have heard of it, there are these go no go tasks. That is, they keep a red and a green button on the screen. If they give green, you have to make a movement. If they give a red button, you have to give a no-go movement. That is, you shouldn't be moving. And they see a lot of no-go activations, even though green is given to them. So this is what happens. This is the overview. That is basically at the level of STN, there is an overall hyperactivity, which we have to suppress or try to suppress at least using DBS. When I mean overactivity in terms of synchrony, very high synchrony, we have to break that synchrony a little bit.
Okay. And direct pathway is inhibited, right?
Yeah, it will be reduced in differential way. Yes, it will be reduced a little bit. Any other doubts? Okay. Then we'll go through the coding sessions. Okay. So these are the two things we shared with you guys. This is the entire code repository we have given you guys, which we will be using today for simulations. And this is the Colab notebook. Basically, this repository is being used in the Colab notebook to run your simulations, just to avoid a lot of code clutter. Again, for today's simulations also, there are like normal notebooks where you can change parameters and run. There are also Gradio files. That is, it's kind of a simulator. As you run it, it will open up a simulator where you can just open the change the parameters and run it. I thought Gradio will be easier for you guys to do it for today, but you can experiment with the code files as well. It's up to you guys. So you can open this file basically. Are you guys on the same page? Can you guys open the Colab notebook? It's named as STNGP Gradio. Are you guys able to open it? We have shared it in the chat box also, as well as in the email also. Maybe I'll share it again. That's not a problem. Yeah, this is the code link and this is the repository link. Guys, can you please open the workshop Google Drive link which we have shared with you guys? Yes. Maybe I'll give you guys a minute so that you'll be able to open it up. Okay. Are you guys able to open this Gradio code? Just run it from top to bottom for now. Just run the entire code from top to bottom for now. I'll wait for a minute. You guys can finish running it. Yeah. Are you guys able to open such kind of a simulator page? Maybe you can let us know either on chat box or somewhere. Thank you. All of the others are also able to access it. Guys, this is the most important part because you have to be doing simulations and fill up stuff. Okay. I'm assuming all of you are able to open it. So basically, I'll just give a brief out of what this code is about and what is it actually doing. Basically, if you look at the code in itself, it might have cloned onto this repository which I have shared with you guys. That is this repository, if you open it, it has STNGP codes. If you open all of them, you can open that STNGP file itself. This is the overall code file where you are going to simulate that 256 256 neurons at STN, 256 neurons at GPE. And all of the neurons are basically your Izikevich neurons. You can actually look at it. These are your Izikevich equations. So we'll be simulating all the STN 256 and GPE 256 neurons. All of them are Izikevich and all of them are solved using Euler equations. This was actually covered in your day one sessions as well. This is basically your Euler update. And at the end of the day, we'll be looking at a couple of parameters once we finish the simulation. What is it? I will talk through it. We'll finish the simulation, then I'll talk about what are the things that it indicates. So it actually clones onto this repository and simulates the entire STNGP system. So this is the interface how you will look at. And today, the most important things we will be looking at is lateral strength STN. Remember, I was talking about it. That is, you keep increasing the lateral strength, it will show PD behavior. And also the striatal currents, ISTR D2 GP. This is the current. So these are the two parameters we'll keep playing around with. And also DBS. If you check mark the DBS, it will run DBS simulation. You don't check mark it, it's a normal condition. Is it okay? So if you just click on run simulation, it takes a while to simulate it. You have to be a little bit more patient since it's a heavy file. It takes a minute or two to finish the simulation. Maybe I'll run the simulation. You guys also can run the simulation with the current parameters as your setting for now. Just click on run simulation in your simulator link. Guys, if you're facing any issues with running the simulator, kindly let us know now. After this, we'll ask you guys to do an activity with which you will be doing it in groups and filling out stuff. Just be a little patient. I know it takes a couple of time. Those who are actually interested in running the original code itself, not on the simulator, also can use this run stngp code. That also has the same code, but without an interface. So you can run there as well. Yeah. So once the simulation finishes, you can see such kind of plots. Are you guys able to see such kind of plots once the simulation is done? Or maybe you can look at my screen for a while and then you can go through the simulation again back. So these are the plots which you will actually come across once you finish the simulation. The first, the green ones are, sorry, the blue ones are the STN outputs and the red ones are actually the GPE outputs. For today, let's just focus on STN for now. Let's not worry too much about the GPE. The first one is basically a single neuron voltage pattern, how it looks. Can you guys tell me, is it called continuous firing or is it a bursting activity? How does it look like? It's continuous, right? It's continuous firing. And the second plot is actually the LFP. Someone was asking what is a local field potential, right? So what we do in terms of code is we take a central neuron and calculate the total currents of all with respect to all the neurons and find the potential of it. So this is how it looks like. The third one is a little important one. It's called as raster scan. Have a look at it or understand it carefully. The x-axis is basically time. The y-axis is neuron. That is, if I take this line, this horizontal line, it's say, for example, some ith neuron, or say it's a 10th neuron. It will tell me where all it has fired. The dots indicates where all it has fired. Can you see it's pretty much random? There is no thing like all of them are firing together or anything. It just randomly fires. Each dot here indicates each neuron firing. Right. And the third one or the last plot is basically the spectrogram. Basically, with respect to time, how does the frequency look like? Here, it's almost very similar. When you actually run PD condition, you can actually see two thick bands present in it, one around some 15 hertz and the other around some 25 hertz. And after these plots, the code also gives you these parameters. The first one is entropy, synchrony, and standard deviation. Any idea what is entropy, all of you? Any idea on what is entropy? It's basically spectral entropy. Okay, maybe I'll quickly cover up what is spectral entropy so that it's easy for you guys to understand. Yeah, the same thing, Manu. Yeah, but here the P is basically the power at the that specific frequency. Intuitively speaking, spectral entropy tells me whether the frequency spectrum is broad or thin. That is, if my frequency spectrum is like this, that is, there is a sharp peak at any frequency, like say 10 hertz, 20 hertz, 30 hertz, then the entropy will be very low. If there is a very smaller thing, or if the spectrum is broad enough, then it will actually say it's a high entropy. Does it make sense? Why do we need to look at it? As I told you already, beta frequency is very important in PD. It shows a very sharp peak around 15 to 35 hertz. So it will end up having lower entropy under PD condition and higher entropy under normal condition. Remember this point, it will be useful for you guys. Low entropy for normal, low entropy for PD and high entropy for normal. Is that okay? Yeah, so that is what this number is actually giving. Your entropy is 0.7, which is actually pretty high. So pretty high as in it's actually higher in general. That is for normal condition, you'll actually see higher entropies. The second one is synchrony. Maybe to tell you what is synchrony, I'll show you an example of raster scan. You look at these two raster scans. Can you identify the difference between them? Distance, it seems. Difference between them. In the first one, can you see at each time point, neurons are very randomly firing. If I consider this time point, there are few neurons firing, few not firing very well. Whereas in the second one, you can actually see all of them fire very well together. Are you guys able to see that? In the first one, there's very random firing. In the second one, there's very like synchronized firing. All of them fire very much together. And there is no firing in between also. So we call this as low synchrony and this as high synchrony. Is it okay for everyone? And what is the behavior? Thank you. And what is the behavior under normal, do you think? Low synchrony or high synchrony? Yes. So under PD, under normal condition, we'll end up seeing low synchrony. Under PD condition, we'll end up seeing higher synchrony. So if you look at this one, since it's normal condition, you're seeing lower synchrony as 0.1. Under PD condition, you'll end up seeing higher synchronies. And what is standard deviation? So basically what we do is we take these spike outputs, convert it into an average output, kind of 4 LFP signals from 4 clusters of STM. Assume that I take an STM, take 4 outputs out of it, and compute the standard deviation across them. If it's higher standard deviation, obviously they are very much random in nature, and obviously it's normal. Under PD condition, you will end up seeing very low standard deviation. Is that okay? I'll tell you the reason why we are computing standard deviation when we go to decision making. It's important there as well. Is it okay for all of you? Are you guys able to finish simulation on Gradio? Okay, I'm assuming that's an yes. So then what we will do is we will do some kind of simulations hands-on so that you will be able to understand the differences and stuff. Is that okay? I mean, we'll put you into separate groups so that you'll carry out some kind of simulations and note down all these parameters. Then we'll have a discussion on how the parameters look like and why do they look like that. Yes, Manuel, I'll give you a full slide as well for that. Don't worry. So right now in this, so basically, you mean all the parameters for the simulation? So the first parameter is time steps, how many time steps you want to do your Euler equation, what is the dt, and when we do the connectivity, we define something called as sparsity. That is how much do we need to connect. Each neuron connects to how many other neurons. Here, I think we kept the sparsity to be 0.1. Then ISTR D2 GPE, which is actually the D2 striatum current that gets into your GPE. The lateral strength, which is the lateral connectivity of STN, the lateral connectivity within the GPE, and how does STN connect to GPE, and how does GPE connects to STN, and what if there are any other external currents that go into these STN and GPE. Bin size is something for analysis part. We use something called bin size. You don't have to worry about it here. And some kind of random noise which we keep adding to all the systems. You can put it to 0 if you want to also. Apart from that, there are also DBS parameters. If you want to run it under DBS mode, you have to checkbox this. Right now, only biphasic DBS will work. Others won't work. So you just click on biphasic DBS, don't change it. And these are the parameters. What is the frequency? What is the duty cycle or pulse width it defines? And what are the two amplitudes? What is the positive pulse amplitude, negative pulse amplitude, pulse to pulse interval? And this three parameters define your spread. Where do you keep your electrode? What is the amplitude at that electrode and at that location, and what is the spread of that Gaussian? Does that answer your question, Manu? Yeah. So I will quickly show you what are your tasks to be done. You remember we said that to calibrate your STN-GPE network, it should show a transition from normal to PD condition. So when I say such kind of a normal to PD transition, I need to check out two things. I increase striatal currents and observe whether it transitions or not, or I increase lateral strength and observe whether it transitions or not. Once we see the transition, we'll consider one PD condition and apply DBS and see what happens to the synchrony. These are the tasks we want to perform today in like half an hour, maybe from now. Is that okay? Once we do that, we want you guys to fill in some kind of a table like this. For each of the tasks, what is the lateral strength you chose? What is the striatal current you chose? Condition, entropy, synchrony, and as well as the standard deviation. You can think of it as standard deviation. Is that fine for you guys? We'll tell you, I mean, there are slides also to tell you which parameters to set up and all. So it won't be a lot of trouble for you guys. That is, if you go to task 1A, I'll tell you what are the parameters you need to set it to, and then what are the observations you need to note down. Basically, we are going to split you guys into two separate groups. We'll have TAs in both the groups so that we'll guide you through how to go through the simulations. Is that okay? All of you? Okay, then should we go through the breakout rooms then? If you have any doubts, you can ask us right now. Otherwise, we'll do the breakout room and we'll actually go through this entire activity. And where do you guys have to fill in? There is a PowerPoint presentation that is shared with you guys in your folder itself. In the folder where we have sent you the workshop details, you can fill in there. Any issues? Can we create the breakout rooms then? This is basically for you guys to know it. Yes, Alvin.
Yeah, we can create the breakout rooms.
Yeah, can you create the breakout rooms then?
Yeah, make 7 per row.
Yeah. We can do 2 breakout rooms. Sorry? Yeah, 6 or 7. You can split it into half. Don't assign breakout room for me, maybe.
Okay. Yes.
Oh, yeah, you assign it. I'll just go into that. It's okay.
Yeah, it's perfect.
Yeah.
Everybody okay with the task setup? You guys can also use this time to just a second. Um, can you Manoj, can you open the uh, presentation that has been shared?
Okay.
Yeah, we have shared a presentation, compounding workshop BG project simulation.
Yeah.
Essentially, what you have to do is, given the importance of striatal T2 current to GP and then lateral strength, you have to, there are like two tasks. So essentially, you have to vary the currents and see the synchrony and variance levels for uh, each of the argument sets. And then in the second experiment, you have to vary the lateral strength in the STN network neurons. And then you have to check the levels of synchrony and variance. And you have to mark it in the table that's been given. So we can
Okay.
discuss the table. Is that okay?
Okay.
Manu?
Values
Yeah. Okay.
within
Yeah.
this. Hello.
Yes?
The by default values within the simulations are for the normal conditions.
Yeah. No,
Okay.
yeah. I mean, once you vary these parameters, you will be able to see the synchrony which is seen in the PD or less synchrony which is seen in the normal conditions.
Okay.
The slides are accessible, right?
Yeah, it's in the view only option.
View only. Maybe you can. It's view only. Maybe you can just create a similar table like that and start filling it.
Okay.
Yeah. You can also use this time to discuss among yourself anything that you found interesting or yeah.
Where is the table, Aditya? It's in the PowerPoint.
Yeah, it's in the Google Slides. Slide number 10.
But my slides are showing that as discussion pointers.
Yeah, now I think it'll come up in like a Say, at first, there are two slides discussion pointers. One is with table and one is just empty. It'll be like this, I think. Yeah, it'll be like this. Is it visible?
Can you put up all of your names in your front sheet of the PPT so that we'll know who all worked on this specific part? Just add a new slide to the top. Yeah, you can just below day 4 write down your names.
Below D4 here.
Click on the day 4 text and double click it. Yeah, do enter. Yeah, just reduce your font size and you can enter your names. Maybe to some 30 or 20, I don't know. Yeah.
Charita,
Yes.
so this simulation that we are doing now
Mhm.
is based on the paper that you sent, right?
Yeah, but slightly modified from that paper. I think they have done a connectivity which is Gaussian. We have done something, I think, or the code I have is with random connection. It's
Oh no,
similar, but might be a
okay.
slightly different one.
Okay, but then so see, I'm a clinician, so
Okay.
yeah, a clinician with a background in cognitive science, and this is a little difficult for me. But I think the paper would help me. So I understand what we're doing. It's just taking some time to understand why are we doing what are we doing. So the paper would help with that, right?
Yeah, it should. So basically, what I work on is decision making. And generally, deep brain stimulation doesn't improve decision making. It rather
Yeah,
does anything with respect to it.
yeah.
Ideally, by the end of the simulation, if we are able to finish it till the end today, you'll actually see the gambling disorders are not really improved with
DBS, no,
DBS.
it's not. Yeah,
Yeah. So the reason can be explained when you do these simulations. You're noting down those the standard deviations, right? When you do the
right,
simulations under normal
right.
PD and standard deviation, you'll actually see that even
Aditya, can you just show the table? Because I am not able to see that in my slides.
Sure, yes. Hope everyone has seen the chat. So maybe one person can share their screen and go through the simulations. Uh, I'll split up the simulations among yourselves so that the work gets done faster.
The simulation processing, it takes a lot of time.
Yeah. So if you can split work amongst yourself, it just need one presentation per this breakout room or one presentation per group. So that the work will be done faster. Just with one person filling up the table. The others?
Can you just show the table once more, Aditya, so that I can see it?
Sure. I'll just share it.
Actually, this gives a standard deviation, right? But we have to enter variance in the table, no? So we have to square it. Huh?
No, yeah, you can enter the standard deviation. Yeah.
Okay, okay.
Click something in the chat. Okay, show. Yeah, 0.276.
I have run the last row. So
Last row.
entropy
Okay, last row was 0.3. Okay, that's fine.
bursting and entropy 0.276. Entropy 0.276, synchrony
Nice.
0.684.
No variance. What is the task to?
I mean, I squared it actually. So
Oh, yeah, okay. There'll be something around 0.06, I think.
yeah.
I ran the first two rows.
Yeah. Which
Uh,
are?
uh, when str underscore d2 is 0.5,
Uh,
so entropy is 0.849
uh,
and synchrony is 0.084
4. Yeah.
and standard deviation is 0.26.
That is, oh, yeah, okay. You would have observed continuous firing, right?
Yes.
Here
In
you would
both
have
the rows, it was continuous.
both the rows.
I think so. I'll check again the second row.
Okay.
When SB2 is
Yeah, I
5.
think it is continuous only. I have increased up to 10, still it was continuous, that current in the same lateral strength. Second is continuous.
What are the values?
Entropy is 0.3,
0.3,
synchrony is 0.698,
0.698,
and standard deviation is 0.06.
0.06. Okay, I think I have to look at the task again.
So these values are for STN, which I told. It's fine, no?
Yeah. Uh, this is the final task. Is with DBS, right? Yeah. What else is remaining?
Task 1B first row, which I'm running right now.
So task 2, we don't need to enter anywhere, right? We just need to run and see, huh?
Yeah. I think the last row, right?
Oh, I didn't. Oh, shoot.
Yeah, this is
Okay.
with. Have you applied DBS?
No. I will run it again. I
Can you
didn't
run
know.
it?
Yeah. It's all error. Just remove it. Huh?
Yeah. Check it. I think you'll get a mix between the continuous. You won't get full bursting, but you'll get a mix.
Task 1B when lat strength is 0.02.
Just in 1B.
1B 0.02 lat strength. It's continuous and entropy is 0.919.
Yeah, okay.
Synchrony is 0.094.
0.094. And
And SG is 0.25.
I have
yeah,
to run
this
the
one is surprising. Huh?
biphasic DBS for task 2.
Uh,
DBS is biphasic, no?
yeah. Only that option is available.
Okay.
What about others? Uh, uh, is it okay? Pooja Velaban and Renu Dipti, are you also running simulations on your own?
Yeah, we are carrying out it.
Yeah.
Hello, Aditya. Hello, guys. Is everything
Uh,
going fine here?
yeah, just running the last task
Yeah, no
with the
problem.
biphasic
Finished.
TBS.
Yeah, I gave break till 11:45 in that room. It's done. Once you guys are also done, you can take break till 11:45. Then we will resume.
Sure.
Yeah. We can remove the breakout rooms by 11:45.
Yeah.
Yeah.
Just waiting for this one to this thing.
Yeah, no problem.
My notebook is running into an error. Just give me 2 more minutes.
Oh, what kind of error are you facing?
Like net connection lost, and then it's not loading. So I'm just
Oh,
rerunning.
okay.
So when DBS is there, we see as entropy as 0.322,
0.322.
synchrony is 0.428,
Oh, how did it get so? Okay.
0.428, and SG is 0.08.
0.08.
Yeah, and the plot looks like busting.
Yeah, it's close. It's a mix. It's mixed between. It's not
Continuous
fully
and
bursting.
yes.
Yeah, that's the effect of DBS. It acts like kind of a noise to the system and delays the uh, firing synchrony. So you get that kind of entropy.
Aditya, just a pointer. I think the second one, 0.0275 for 5 will be bursting.
Which one?
Yeah, that one will be bursting. The
Yes,
synchrony.
yeah,
Look at
I
the
have that
synchrony.
the same doubt, but someone said that it was continuous. So
No, it was bursting. We ran it there also. Maybe you can rerun it and keep the result. I have the result
yeah,
for it. I can show it.
yeah. I've also checked yesterday, but right
No,
now I've
we
just
ran
kept it.
it till now only, but it'll be like bursting. Yeah. Yeah, need that. Task 2, can you put it as DBS something so that we'll know that it is a DBS thing? Forgot.
Yeah.
Yeah. Okay, just maybe quick pointers for everyone before you go for a break for 5 minutes. You can see continuous is normal and bursting is PD. You can write it also after continuous in brackets as normal and bursting as PD. Can you guys observe that as the striatal currents are increased, you can see that it goes from normal to bursting, that is normal to PD. Similarly, in the second one also, when there is from low lateral strength to high lateral strength, it goes from again normal to bursting or normal to PD. Okay. But if you actually look at the metrics itself, under normal conditions, what is the approximate synchrony you guys are seeing? Can someone put it on your chat box or unmute and mention it?
Sorry, come again, please.
What is the approximate synchrony you see under normal condition? Some 0.1, because in normal condition there are two normal conditions and both of them the synchrony is 0.08 and 0.09, right? Can I approximately say synchrony is low around 0.1?
Yes, yes.
Same for PD, it's approximately 0.7 in a way. It's 0.69 and 0.62, right? 0.7 is the synchrony, approximately. Now, on applying DBS, what happened to the synchrony? Did it reduce or not? Under PD, it's some 0.7. Under DBS, it's some 0.42. So it reduced a little bit, right? It did reduce synchrony a little bit. Can all of you observe that one point? Yeah. And second thing you guys have to look at this is also in terms of variance. Even though the synchrony reduced, did it reduce as much as normal condition? Not really. Synchrony of normal is 0.1. For PD, it is 0.7. DBS is 0.4. It didn't reduce till normal. It reduced to some extent for sure, but not restored till normal. Second thing, can you also look at variance? Did DBS increase variance a lot? In PD, it was 0.07. Under DBS, it's 0.08. Did it increase a lot? No, right? Under normal condition, my variance was 0.25, whereas under DBS, it's only 0.08. This is actually a very important point when you go to decision making. Keep a note of it that DBS does reduce synchrony, does improve a little bit, but it doesn't actually increase variance a lot. Have this in your mind because we'll be doing decision making simulations in the second half of the session where you might actually need it. Is that okay? Yeah. Okay. Then why don't you guys
I
go for
think
a quick 5-minute break? Yes, Aditya.
no.
Sorry.
I just say we can end the record rooms, but okay.
Yeah, we'll end the breakout room. We'll take like 5 minutes break. We'll resume back from 11:50. Is that okay? Everyone kindly go for a break for like 5-10 minutes, then we'll be resuming back. Yeah. Aditya, can you close the breakout rooms? Hello everyone. We'll be starting by 11:50. So guys, let's start now. Should we resume back? Are you guys back? Can you let me know? Okay, cool. I know it's very intensive like that, but yeah, anyways. Okay, so we'll get started now. So the second part of the session will be giving you a brief overview on how do these ST dynamics make an influence on decision making. Okay, maybe let me share my screen first. Yeah, can you guys see my screen? Is everyone able to see my slides now?
Yeah, screen is visible.
Yeah. So in the previous exercise, all of you might have done this exercise, right? You might have done normal PD, and PD DBS conditions, and you would have seen the difference between normal and PD and how does DBS improve, right? Now, as you guys might have observed, that standard DBS does improve to some extent, but it is not as good as normal condition. And just have a note on these standard deviations as well. That if you actually look at the standard deviation under normal condition, that is around 0.26 or approximately to 0.2, 0.25. Under PD, it's somewhere like 0.06, 0.03 stuff. But as you do DBS on it, it will slightly increase, but it doesn't increase it a lot like in normal condition. I'm stressing on this point because when you do decision making, you will understand why the standard deviation makes a lot more difference when you do the exercises. And these are the conditions how you might have observed, or the plots you would have observed. Under normal, you would have seen such kind of a continuous firing and there is no specific peak that comes up. Whereas under PD, you will see that there is a bursting condition and there is a strong beta peak. As you do DBS on it, the beta peak slightly merges off. It doesn't show a lot of strong high power in those peaks, but still it is not very well. Okay. Now going to the second part of the session or the project, which is decision-making deficits in Parkinson's. Apart from motor symptoms, decision making is also impaired in Parkinson's patients. When I say decision making can be in different ways. It's, as I mentioned before, also it's very integral for daily life. And what are the actual deficits you end up seeing? The first one is impulsivity. They show a lot of gambling disorder and they end up losing a lot of money in gambling. Second is risk-taking behavior. If you give them some kind of a risky choices to make, that is put on a high risk stock versus a low risk stock, they end up preparing or preferring a high risk stock, that is the risk taking capabilities are very high. Their understanding on reward, loss or punishment or probability of rewards is very very bad. They can't really show a lot of cognitive flexibility. When I say cognitive flexibility or set shifting, that is, I give them certain kind of a task to do and suddenly change the paradigm of the task. They can't adapt very easily. They have difficulty in adaptability as well. But these are a lot more decision making aspects. We are not going to look at all these decision making aspects. Maybe we'll only look at one kind of gambling issue, that is Iowa gambling task. Iowa gambling task is very well studied under PD patients for a very, very long time. I'll talk about the task also, and I'll also talk about the framework with which we will be looking at. That is, to understand this decision making, how do we make decisions? We should be understanding the reinforcement learning perspective of basal ganglia. Any idea what is a reinforcement learning thing means? Like, what does it actually mean? Where is chat box? Why can't I see it? Yeah, any idea? Anyone?
So you basically try to increase a behavior by giving some sort of a reward.
Yeah. So basically, this is how we say generally people learn. It's a reward-based learning. The way we humans tend to learn is we keep doing the same activity or same actions which we find are rewarding. Like studying before the exam. Might be rewarding to few people, might not be, but still. Generally, that's how we learn. So we have to understand a reinforcement learning perspective. But before that, let's just understand how do we make decisions. Say I have a couple of stocks which I want to invest in. How do you decide where to invest? Maybe let me open the chat box. Yeah. How do you technically try to define where to invest in? What are the things you will do? It's open for discussion. You guys can let me know. What do you guys think? If I have some money I want to invest in, what are the things you look at and what are the things you will do to invest? No one has invested anywhere.
We look at the trends and what holdings they have, what companies. We look
Yeah.
at the past year's trends.
Yes, you're right. So one thing we will do is we will try to look at some kind of a price history of it or estimate some kind of value, which will tell me this stock is good or this stock is bad, right? That is one thing I'll do. And look at the best possible stock based on the price history and invest in it. Do we always tend to invest in the best one only, or do we also try out other stocks thinking they might be beneficial? What do you think? We might not go for the best thing all the time. We might try to see other stocks also, right? Because they might actually give better rewards in future. Yes. So these kinds of behaviors can be classified into explore and exploit. Let me go through it in a little more deeper aspect. Yeah. So basically, what do we do is we will see which of the stock has consistently performed better in terms of returns. They might have gotten some returns in the past, right? Which might have performed better, and we try to pick it up. Or we also try to pick out other stocks. We might not really go and pick only the best stock. We might also try out. It's very human behavior that we also tend to go ahead and put in some other stocks thinking they might also be better, which is actually explore. Exploit is where I know this is the best choice possible and I'll try to pick it up. Explore is I might not know the options very well, which or the choices very well. So I'll try each of them and see which one might be better over the time. Does that make sense to all of us? So any decision making will have these four parts, that is explore, exploit, and the second is value. So the same explore, exploit can be explained in terms of restaurant picking. I mean, in town, I know there is a restaurant A, which is best, which I have been eating for a very, very long time. So I'll keep going to the same restaurant. But at times, I might also explore other new places. Can anyone suggest why is this exploration very important to us? Why is exploration very, very key to us? Yes. If I don't really explore the other choices available, do I know which is the best possible for me? Because I don't know really which is the best one possible. I have to try out a couple of new things to understand which is best for me, right? So don't you think exploration is very, very important? And exploration is pretty random as well. By that, I mean, when I have n choices, I will randomly make a choice in exploration. Exploitation is where I know that this is the best thing that is possible and I keep doing it. That is the first thing that is a very key aspect of decision making. And the second aspect of it is value. We'll always look at an average reward or an average return that this choice is going to give me all the time. That is what is value. Like here, the average return I might have gotten in the past is what is value. The same thing is value here. So these two are very, very important components in decision making, in any kind of decision making. And they're very predominantly used across all the RL literature as well. Any doubts still here? Or if anyone has any queries still here, because this is the important part of the next topic. Any doubts still here? Cool. I'm assuming there are no doubts. Let me go ahead with it then. Cool. So, but in terms of basal ganglia, we know that we look at the entire architecture as go, no go pathway. Where do we have such kind of a decision making capability? Because basal ganglia is known to be a reinforcement learning or a reward based learning engine for the brain. Where do we actually end up having such kind of a learning? For that, we have a hypothesis that the entire exploitation goes through the go pathway and the exploration comes through the no-go pathway. That is, direct pathway has this value which it keeps computing in its input, saying that, okay, fine, for this choice, probably this is the average reward I have gotten. It keeps computing it at its D1 pathway. And D2 or the indirect pathway actually gives you that randomness to explore different choices. Now, when I say randomness, the randomness actually comes from STN-GPE system. So the STN-GPE system is what is very, very important for exploration. When I say randomness, can I also say that higher variance, higher randomness? Yes, right? Higher the variance or higher the complexity, I can say higher the exploration. That is what we are writing here. Generally, the classic go, no go view might not be the key to look at the entire decision making problem. We have to actually look at it in terms of reinforcement learning framework, where we look at the direct pathway as exploit pathway, where I keep estimating the value and say, okay, fine, this is the best choice I can make. And the exploration in itself comes from the indirect pathway. And that too, it comes from STN-GPE system. Higher the complexity or higher variance at STN-GPE system, there will be higher exploration. Lower complexity, we will have lower exploration. Now, under PD condition, what do you think happens to the complexity or the variance? Can someone type on your chat boxes? What happens to the variance under PD condition? Or the variance that is at STN-GPE? It is very, very low, right? It is somewhat like some 0.06. Yes, ma'am. Whereas under normal condition, the variance is pretty high. Can I say the exploration is what is actually deteriorated under Parkinson's at the level of indirect pathway? There's also more issue, but this is one thing that is deteriorated. Yes. Apart from that, can I also say that value learning might also be deteriorated? By that, what I mean is, see, if I want to keep estimating the value, there should be some kind of training that I have to do to the network. Yes, every time I have a value that I have estimated. See, currently I have a value of some V that I have estimated for a task, that's for a card or for a stock. Now, if there is a sudden change in the value, do I need to update the value? Yes, I need to update my estimate of the value, right? So that will give me some kind of a loss or an error which I have to update. So that is what is called as TD error or a temporal difference error. Let me show you the equation of it. So as I said, we'll always have value that keeps getting computed at the D1 output or the direct pathway. And every time, whenever I compute the value, I'll always compare it with the true returns and create an error. R is the true reward or the true return I got, and Q value is the value estimate I have done. Okay. Now, if I take the subtraction between them or the difference between them, it will give me an error. Now, this error is what we technically train the network with. I'm not going into the full details of the BG network. The BG network is actually a simple neural network itself. All of them are neural network layers, like independently, all of them are sigmoid layers. And STN-GPE can be your spiking network. And the error we use to train the network is actually this TD error. Now, this TD error is very important because this temporal difference error is what correlates to dopamine in brain. Whenever there is this error, dopamine spikes. Now, this also gets depleted under PD condition. Now, my question to all of you is, if my error is not correct, if my error in itself is depleted, what do you think will happen to the learning? Do you think my value estimation will be correct? If the error estimated itself is wrong, do you think my value will be correct? Yes or no? No, right? If my errors are higher and higher, obviously my value won't be learned. I mean, error is wrong, then my value also won't be learned. So under PD, if you want to look at what happens to decision making and why is it impaired, it's because of these two problems. One, they can't learn the value itself correctly. And the second thing is exploration is what is lost. In PD, this is the reason why any cognitive task doesn't really work well, because of value impairment, that is, they can't estimate value, and their exploration is lost. Now, make a quick guess on your chats. When I say DBS is given to STN, what do you think it's trying to do? Do you think it should be targeting exploration, or do you think it's targeting your value? Exploration, right? Yes. So basically using DBS, we can only restore exploration. That also is not fully restored, because you see that the variance is not very, very high. Value learning is also impaired, but for today's session, I'm not touching the value impairment in itself. Okay, it is also impaired. So only in few conditions we can actually use DBS to improve decision making. Otherwise, we can't really improve it. That too, with standard DBS, you will see it doesn't improve. Are there any doubts still here? If not, I'll go through the task paradigm and then tell you what are the simulations you can do and observations we can make. Okay, cool. So
what is
yeah.
the cause of value impairment in Parkinson's?
So Manal, you have learned about these neural networks before, right? So the entire training happens through loss function. Yes.
No,
If
I mean in Parkinson's disease, actually, in humans.
Yeah, the same thing I'm saying. So when the dopamine is lost, that is, this error is the clipped off, right? See, when my true value and the current given reward are not matching, dopamine should fire, saying there is an error, update your value. Does that make sense? When these two, the true and the estimated value, are not matching, in normal brains, TD error will be high. And the dopamine cells start firing, saying there is an error, update your value. But in PD, since the dopamine cells died, this error won't be updated, or your value update won't happen, because of which your value is still impaired. Does that answer your question?
Okay.
Yeah. So going through the task paradigm. So this is one of the classic tasks people use. It's Iowa gambling task. It's kind of, you can think of it like a low risk versus high risk stock. What they do is they have 4 decks of cards, A, B, C, D. All of them have certain reward and loss they will give it. A and B cards give high rewards, 100 and 100, but their loss is very high. If you actually look at the average across some 10, 20 trials, you'll see that A and B cards actually lead to loss. Whereas C and D cards, though they give you lesser rewards, they're actually rewarding over time. This is what Iowa gambling task does. Okay, so we will technically look at, generally patients are made to play this task, and they observe that under normal condition, C and D cards are more preferred by people, whereas under PD condition, A and B cards are more preferred, or they will tend to have lesser scores. They are not very good at distinguishing these two. Is that okay? So what they do is they compute something called as an IGT score. That is, how many times did I pick advantageous cards minus how many times did I pick the disadvantageous cards. That tends to be higher in normal and lower in PD. Is that clear to all of you? Yeah. So since we have only 20 minutes, we'll be doing one simulation for today, and the other you can do it as your on your free time, actually. So the tasks are these. The entire basal ganglia network integrated with STNGP is already present to you guys. Okay, all we need to do is do simulations for 2 times. One where we will just remove the entire STNGP system and say that it's just a random noise at STNGP it is generating, and try to do the simulations. And the other, you can actually integrate the overall detailed spiking network and run the simulations. Both of them will show you very similar results, but spiking network takes a lot more time. So maybe since we only have 20 minutes, we will do the noise simulations. That is, by noise, what I mean is at STNGP, instead of having a spiking network, I'll just have a simple random noise with some variance that is present. Is that clear? Any doubts with respect to why we are doing it and all? As I already mentioned, STNGP system is very important for generating such kind of a randomness. We actually can replace it with a random noise and try to test the network in itself. Okay. Now, what are the variances can we see? Do you remember when we did our STNGP experiments? Do you remember we also computed these standard deviations? We can test at these standard deviations in itself. That would be the fastest thing to test whether the IGT will work or not, right? Is that okay? Okay, so maybe then we will go through the simulation in itself and understand how the task will be varied. Okay. For that case, you guys have to open your Gradio code for IGT. Are you guys able to see the Gradio code for IGT? This is the IGT Gradio code. Can you guys run it from top to bottom? Just simply run it from top to bottom. Yeah. Once you run it, it opens a Gradio page like this. Are you guys able to see that? Can you let me know once? Yeah. So what we will be doing is we will be doing either noise experiments or STN-GPE experiments. For now, don't use the STN-GPE because it will take a lot of time. We can have different variants at STN level and run the simulations. Is that okay? That is the task. We will run it for normal variants, PD as well as DBS. I'm sure you would have remembered that under normal condition, we had certain variants at STN. Under PD condition, we saw certain variants at STN. And under DBS condition also, we saw certain variants at STN. We'll run it for those variances. Is that okay? Okay, for normal condition, what was the variance we took? Approximately. What was the variance we saw under normal when we simulated STN GPE network? Somewhere like 0.2, 0.25? Yes? Yeah. We can run it with that number then. Just to have a brief overview, you can actually use the entire spiking network. It will give very similar results. Yeah, just run it up. It might take a while, but just be a little patient with it. Yeah. Once you run it, you will see something like this. One is the IGT score and how many times each card has been picked across the different trials. I mean, they make them pick for some 100 times and you will see it over time. How does it vary? Can you see that the scores are actually positive in its end of the trial? That is, by the end of the game, they understand that what are the advantageous cards and versus what are the disadvantageous cards. If they pick a lot of advantageous cards, then it's positive. If they pick a more number of disadvantageous cards, then the scores will be negative. So can you see under normal variance, the values are pretty much good? That is, they are actually able to learn it. Similarly, you can run it for PD variance. What was PD variance? Somewhere like 0.04, 0.06, right? We can run it at those variances. You guys also can do the simulation yourself and test it. Let me put the numbers also. Yeah. Now, when I put a variance of 0.04, do you see that the scores are very worse? That is at some -5, -7. Can all of you observe that? That is, the scores have gone negative. Similarly, you can also run it for the slightly higher variance.
Ma'am, I'm putting the same values, but the plots are looking different in your case and in my case.
Yeah. So basically, see, when I versus you play the game, it won't be the same score every time, right? It will have person to person variability. So when you
Okay.
simulate it, the noise at STN-GPE will take a play. You have to only look at overall if the score was positive or not. And then you can do a statistical analysis actually to prove that DBS and PD scores are not statistically different. So generally, we tend to run it for like some 10 to 20 times separately and see the scores. I mean, one simulation might not give you the exact value, but in general, when you actually run it for lower variances, the scores are negative.
Okay.
Yeah. At least these are the scores I am getting right now. You can also test it. Yes. These are the brief scores I am getting right now.
So,
Are you guys?
like, is the same code running? So,
Yeah.
how is it creating individual variability within the same code? Like, is there some library which is used that uses randomness every time you run?
Yeah. So whenever I run my STN system, it takes it as random noise, right? So the value is a random noise there. So the randomness comes from that. It takes at np.random.random. If you actually open the main repository and check it, you can see that. Even when you run it with the spiking network, okay, you see that there is a variance within itself, even in the spiking network, right? Because if I take some random time point in the spiking network and start my simulation from that time point, obviously it will be different from the other person, right? Because my randomness might not be the same at SDN with respect to the other person at that time, or the same number might not come up. That is how people make different decisions, and exploration is always random.
Okay.
Yeah. And
And then there
technically,
in the
yeah.
below the figure there is 0%, 0%, two rows. Like there are no mention of steps or anything. I don't know, like
Uh,
in your case it is not visible. Right below it there is.
Here,
Below the IGT metrics there are
uh,
steps or something coming in my screen.
steps. Can you share your screen? I don't know what is happening though. I can stop my sharing for a while, or you
Oh
can
wait.
pin the image of it.
Wait, I'll show.
Yeah. So we don't need to pin PD, no rhythm. Actually, if you are running the ST and GPE system, then only you can use the drop down. Otherwise, you don't have to, because here variance in itself is determining the PD condition, right? For ST and GPA to load the spiking network parameters, that drop down is there. Otherwise, that drop down doesn't make any sense. Yeah. It might also be some rendering issue, Varnika. If you can reload or rerun the system, maybe it will work sometimes.
Okay.
Yeah. Yeah. All of you are on the same page till here. Does anyone have any issues till here? Okay, now maybe I'll just open it up since it's only like 10 minutes for the discussion. Maybe I'll open it up for the discussion as well. Why do you think so? Under 0.025 is normal variance, right? This is the variance we got under normal condition. And this is the variance we got under PD DBS. And this is the variance we got under PD condition. In which condition the IGT scores are positive or higher IGT scores? Of course, under normal condition. Under PD and DBS, do you see any much difference in terms of IGT scores? Like, did DBS improve the IGT score in any sense to a lot of extent? Like in normal condition, you see? Yes, I have put up the values on the shared screen. You can have a look at it. I can't look at the chat. No, right? Why do you think so? Oh, I think it's a rendering issue, Varnika. I feel it's a rendering issue. You can refresh all of it and try it out. Yeah, it's just a progress bar. I mean, in my case, it shows on the thing itself and goes off. But I think in your case, it's showing the rendering bar there.
Okay, okay.
Yeah. Or you're using it within the VS Code, within the Jupyter Colab notebook. You're not opening the browser itself.
I'm using the collab only, but
Oh, okay. Then you have to scroll within it. Or you open that public link, or you open that localhost link and do it.
okay.
Yeah. Yeah. Okay. Just going back to the question I posed. Why do you think that DBS didn't improve the performance a lot? At least from whatever we have observed till now and understanding of the RL framework. Why do you think DBS might not have improved the performance a lot? Because it didn't improve variance a lot more, because of which the exploration is not really very well restored. And due to that, can I say DBS wasn't able to improve the performance a lot more? Yes. Maybe I'll stop my screen share. Yes. Don't you guys think that? Why can't I see? Yeah. So that is what, or at least that is the hypothesis with which we think that the DBS wasn't able to improve the performance a lot more, because it doesn't really restore the exploration. Though we haven't touched the value impairment yet, we can also impair a lot of value, like to a greater extent, and then see that DBS can't improve irrespective of whatever it is. But one reason why DBS fails is it's not able to restore the entire exploration that is needed for decision making. That also makes sense because if you look at PD patients, they're not good with changing environment. Like I give them certain task paradigm and change the task paradigm, they can't adapt very easily, because whenever there is a task change, then you have to explore more to go back to the optimal performance. PD patients can't do that. So the current TBS protocol, even though it's actually good for tremor, but it is not really good for decision making in itself. And to improve for decision making, we have to come up with certain kinds of protocols which can actually remove the entire synchrony of and restore the dynamics as it looks in normal condition, which I think DBS doesn't really do, which can be a research problem statement in itself you can possess further. Is that fine? Or do you guys have anything that you want to add on or ask something? You guys can also simulate it with STN-GP system, just that it takes a little while, but it will run.
Can you please summarize the trend? I want to note it down.
Yeah. You want the full trend, or if I show the last bin score, that's enough, right?
Yeah.
Yeah, wait, let me share my screen. Yeah. Can you see my screen, Varnika?
Yes.
Yeah. So this is what we have done in terms of IGT. We have taken the variance at STN, which we saw when we modeled the entire spiking network, and ran the IGT code with that specific variance. Okay. That is, under normal condition, the variance was 0.25. PD DBS, it was around 0.07, and PD, it was around 0.05. And we ran the code. And here I'm just giving the values of last bin IGT scores. Under normal condition, the score is positive, implies they have picked a lot more advantageous cards in comparison with disadvantageous cards. Whereas under PD and PD DBS, the scores aren't really positive, they are still negative. That is, DBS doesn't very well improve. If you actually run it a couple of times and do statistical analysis, you'll see that both of them are not any more statistically different. Does that summarize for you?
Yes.
Yeah. To be more specific and to give out the pointers, I think, wait, let's open this itself. Yeah. So these were the pointers I thought you guys can actually have a look at it. So basically what happens is when you keep increasing the variance at the STN level, you will see that there will be a point at which the scores go from very bad to very good. That is the distinguishing point. So normal variance is pretty high in such a way that the performance is good, or they explore enough that the performance is good. Under PD or DBS, the performance is bad because they are not really able to explore much. The same thing you can see under normal PD and DBS as well. Does that answer all of your queries? Do you guys have any more queries you guys want to ask or you want to discuss here?
I have a
Sabha,
doubt.
yes, Mana.
Suppose we want to really measure like these variance values in STN in like human beings. So apart from like directly measuring during the time of surgery, does any investigation like fMRI or EEG help us to measure these things?
No, because since they are very subcortical in nature, the superficial brain structures might not be able to capture it very well. Though people have been trying to study how can they use fMRI and stuff. I mean, till date, people are not able to classify PD properly because they can't know if actually the cell death has occurred unless and until they go into the brain. Like just imaging might not be enough. That is one of the reasons why there is a lot of misdiagnosis also. So it's not very straightforward.
Okay, so like there is no other way other than simulations to actually model these things, right? So we
Yes.
cannot.
I mean, experimentally speaking, unless and until you actually do cut open the brain and look through it, it's not possible. So one thing which Medtronics keep doing is they have this feedback stimulator where it also keeps recording. So when we implant electrodes for stimulation, you can also have recording electrodes with it so that the brain signals can keep recorded also in parallel. But the only problem is it affects your battery life because recording also takes some energy, right? And that has to come out from the patient's pocket, which no one would be ready to do. So the amount of data we get is also from the literature or the research data, but not really from the clinical data. At clinical level, it's very difficult to get it.
Okay. Or like they should do some animal models or something, right?
Yeah,
And then we
animal
can.
models are there. Yeah, they do a lot of animal studies with it, but then it's not very easy. And also decision making task and all, they do it with humans only. I mean, humans who are actually implanted with electrodes. But again, when they do decision making task, they don't have recordings that are submitted with it. Very few papers actually submit the recordings of STN also along with the data they record with decision making. So correlating both of them will be very difficult. That is the reason no one is right now able to pinpoint why should DBS work and why it shouldn't work for certain paradigms. So modeling becomes a tool to understand why at least it should work or not.
Okay.
Yeah, I mean, especially at the clinical level, no one really understands yet why should DBS work or not. That is the current scenario in terms of experiments, at least. Modeling at least gives some insights, probably. And Sabha, I think that answers your question. Why do you need it and why is it important to look at STNGP dynamics in itself?
Yeah.
Yeah, let me know if you guys have any other questions. I mean, one further study, as I said, is to look at how can you have deep brain stimulation protocols to improve decision making. That is the further point of view to look at it. Okay, then if that's all the questions you guys have, maybe we will stop here for today. If you guys have any queries, you can reach out to us via email. I'm sure you guys have our emails. You can reach out to us if you have any questions regarding the simulations or anywhere you're stuck, you can let us know.
Just one more doubt.
Yes, ma'am.
So can't fMRI just measure this, like say, for example, thalamus, say STN thalamus or thalamus cortical ball fluctuations, and kind of give some measures to study all these things? Or is it
Yeah,
not accurate enough?
it might not be accurate because blood supply in general, I mean, if I'm not wrong, they look at the blood oxygenated levels under fMRI. STN firing under normal to PD condition, once the PD has already attacked, how do you check if there is any change? It might be already overactivated, right? And if you do also, what is the accuracy that I can predict it with? I don't know. I mean, people have been actually trying to see if they can capture the same beta activity near the EEG, but they aren't very successful with it. Even with EEG, people have been trying to see if EEG alone is sufficient. Can I characterize PD using EEG? But they are not very, very successful with it.
Okay.
Yeah. If there's any very recent literature which they might have, I have no idea. This is something like a year or two before I know. Currently, I have no idea. Okay, then that is it for today. I hope you guys were able to understand at least few parts of it and understand the simulations to some extent or have an overview of what is basal ganglia architecture and how does STNGP work around. With this, I'll be closing the project session for today. Kindly contact us if you guys are having any queries. Thank you guys. Thank you so much.
Thank you.
Thank you. Aditya will close the session.
Yeah, we'll close the session.
Yeah.
Thank you, everyone.